Good day, dear students. Topic of my today lecture is endocrine system. Endocrine system in our body regulates metabolic activities in our organs and tissues, thereby helping to bring about homeostasis, produces a slow and diffused effect via chemical substances called hormones, which are released in the bloodstream to influence target cells at remote sites. Organs of the endocrine system can be subdivided into central endocrine organs and peripheral endocrine organs. Peripheral endocrine organs include pineal body, the hypothalamus, and the pituitary gland. Peripheral endocrine system has three components. First of all, it is ductless glands. Glands which lack system of excretory ducts. For example, thyroid gland, the parathyroid gland, and the suprarenal glands. The second part of the peripheral endocrine system is distinct clusters of endocrine cells within certain organs of the body. For example, it may be islets of Langerhans in pancreas, endocrine cells in sex organs like testes and ovary, and some other organs. And the third part of the peripheral endocrine organs is Apud system, abbreviation of the four capital English letters, Apud system. It is endocrine cells in the epithelial lining of the digestive tract and in the respiratory system. It is system of single cells, and these cells are classified according to the capital letters of the English alphabet. Today, during the lecture, we will discuss only some certain central endocrine organs and ductless glands as part of the peripheral endocrine organs. Distinct clusters of endocrine cells and Apud system we will study during the studying of this concrete system of organs or organ. So, the main secretory product of the endocrine cells we call as hormones. Usually, hormones released directly to the bloodstream by the merocrine secretion. Hormones are molecules with specific regulatory effect on the target cells, tissues, or organs that is often located far away from the gland. Hormones elicit specific and dramatic effects at very low concentrations. They can regulate carbohydrate, protein, and lipid metabolism, mineral and water balance in the tissue fluids, growth, sex-regulated difference in body shape and sexual functions, and behavior, temperature, and emotions. We think that endocrine system, it is the second main regulatory system in humans and animals' bodies. Now, the first place you remember, take the nerve system. But nerve system has rapid influence on the tissues. They produce and carry electrochemical impulse or secrete special substances like neurotransmitters. Hormones provide the long effect on the organs and act for a long time, but effect is not rapid. It needs some time when the hormone secreted then flow in together with the blood and then find its target cells. By the chemical nature, hormones are classified into three types. First group, water-soluble hormones, proteins, and polypeptides. These hormones, example of these hormones is insulin, glucagon, and follicular stimulating hormones, maybe some other hormones. The second group, also water-soluble, is amino acid derivatives. It may be modification of one amino acid, mostly it is tyrosine and hormones example, tyrosine and epinephrine. And the third group constitute the hormones which are lipid soluble. This group, it is steroid and fatty acid derivatives. Examples, progesterone, estradiol, and testosterone, and some other hormones, not only this. How hormones function, or what is the mechanism of action of the hormones? As the hormones released into the bloodstream and has arrived in the vicinity to its target cell, it first binds to specific cell surface receptor or on or in the target cell. The binding of hormone to its receptor communicates a message to the target cell, initiating signal transduction, formation of cyclic adenylate cyclase. It is a special reaction, a pair complex, cell surface hormone plus cell surface receptor to hormone plus hormone, and this complex stimulate the changing in the biochemical reactions in the cells. It means the translation of the signal into the biochemical reactions. This mechanism is true for all types of hormones, but mostly for water-soluble hormones, of first two groups. If we will talk about the lipid-soluble hormones, we can see that formation of complex cell surface receptor plus hormone, which stimulate the production of cyclic adenosine cyclase to synthesis cyclic adenosine monophosphate, it is only the first step. Then this complex stimulates protein kinase A in the cytosol. In such an instance, cyclic adenosine monophosphate acts as a second messenger. This second messenger binds to specific binding proteins in the cytoplasm and the nucleus. These proteins subsequently bind to DNA and directly affected gene transcriptions, which change protein synthesis in the cell. That's why you can understand that the influence of the water-soluble hormone or steroid hormone is not easy. It changes the entire process in the cell, protein synthesis, and has very deep influence on the cell and its activity. Examples of several additional secondary messages include cyclic guanosine 3,5 monophosphate, CGMP, metabolites of phosphatidylinositol, calcium ions and sodium ions in neurons, and some other, of course. One more interesting position, that our endocrine system functions on the base of the feedback mechanism. And this feedback mechanism regulates the endocrine gland's homeostasis. This feedback mechanism functions between the central and peripheral endocrine organs, and also between the peripheral endocrine organs and target organs or cells. It can be negative and positive. Once a hormone activates its target cell, an inhibitory signal is generated and by this target cell and returned to the endocrine organ. It is negative feedback mechanism. When the hormone level is inadequate to elicit a sufficient metabolic response in the target, a positive feedback mechanism takes place. That's why a positive feedback signal travels to the endocrine gland and initiates an increase in hormone secretion. What does it mean? On practice, it means that if person use the hormones of the endocrine gland as tablets, for example, for treatment of some pathologic situation, cells send the negative feedback mechanism signal to the organ which produces this hormone. And hormone and gland decrease or stops the production of the native hormones. And if person use these tablets, these hormones for a long time, it may be even atrophy of the native endocrine gland. That's why the prescription of the hormones to the person have to be controlled by the doctor very careful, and it is impossible to take away these tablets suddenly. If person use the high dose of hormones, it is impossible to stop the eating of these hormones immediately. Person have to slowly decrease the concentration of hormones from the tablets in his or her blood. Many centuries, doctors think that the most important central endocrine organ is hypophysis. And now we think that hypophysis is the most complex of the endocrine glands, but it has subregulation from hypothalamus, anterior hypothalamus, which control the activity of the hypophysis. And hypothalamus secretes so-called stimulating or inhibiting hormones for almost all hormones of the adenohypophysis. But today I'll retell in details the composition of hypophysis, and it is very important for understanding of the correlation between the central endocrine organs and peripheral endocrine organs. So, hypophysis is divided into two major parts. First, adenohypophysis. It is glandular portion, consists of mostly of the epithelial tissue, and it derived from oral ectoderm that migrates dorsally as Rathke's pouch to surround partially the neurohypophysis, nervous portion. That's why hypophysis constitute the adenohypophysis and neurohypophysis. This slide shows you the scheme about the development of adenohypophysis, green, and neurohypophysis. So, first of all, appear the surface ectoderm, green, which forms the invagination of the sheet of epithelial cells of the ectoderm. This invagination become deeper and longer, you see, deeper and longer, and now we call it as Rathke's pouch, walls of the Rathke's pouch and cavity of the Rathke's pouch. This Rathke's pouch penetrates deep in the cavity of the scale and meet the outgrowth of the floor of the diencephalon. Yellow it is floor of the diencephalon. It forms also outgrowth, or so-called infundibulum, and they meet one another. Rathke's pouch and outgrowth of the diencephalon meet one another and appear organ-like combination of the components of the Rathke's pouch and infundibulum of the diencephalon. And now you see that green, it is adenohypophysis, walls of the Rathke's pouch forms the pars distalis of adenohypophysis, pars intermedia, and pars tuberalis, and also state the rudimental cavity of the Radke's pouch. In human beings, this cleft is very thin and not visible on the simple light microslides. But in mammals, this cavity may be comparatively large. It is a cavity between the pars distalis of adenohypophysis and this thin pars intermedia. Outgrowth of the diencephalon yellow forms the neurohypophysis. You can see it, or pars distalis, or pars nerosa, and it lies opposite to the adenohypophysis. That's why together hypophyses consist of green adenohypophysis and yellow neurohypophysis. Pars distalis, as you see, it is the largest portion of the adenohypophysis. Pars intermedia is now yellow, very thin, and pars tuberalis. Hormone active is pars distalis and pars intermedia. Neurohypophysis has interrelations with hypothalamus. This green, it is hypothalamus. And between the neurohypophysis and hypothalamus forms the hypothalamo-hypophyseal tract. You can see that hypothalamus, it is part of the brain and consists of multipolar neurons, and concentration of the body of these neurons forms nuclei, paraventricular nucleus and supraoptical nucleus. Axons of these neurosecretory neurons travel down to the neurohypophysis, you see, and forms the hypothalamo-hypophyseal tract. This picture shows you the hematoxylin eosin picture of the hypophysis. Dark staining, it is adenohypophysis, which develops from the ectoderm and consists of epithelial and connective tissue. Also, hypophysis is pars adenohypophysis is covered by the thick connective tissue capsule. Neurohypophysis is pale staining. It is a derivative of the nerve tissue. No, of course, have any connective tissue capsule. And you see the very thin pars intermedia. It is hypophysis of the man. You can't see the cleft between the pars intermedia and pars distalis. Around it is the section of the scale. Now pars distalis of adenohypophysis constitutes about 75% of the hypophysis and is enclosed, as I said you, the most completely dense fibrous capsule. The parenchyma of this paradistales is formed by the sheets of epithelial cells. You see, sheets of epithelial cells. On the cross section, these sheets of epithelial cells looks like lobule of the exocrine gland. You can see like the section of the portions of the endocrine gland. Around is the loose connective tissue. It is delicate reticular fibers and a lot of fenestrated capillaries pass in this connective tissue around the sheets of epithelial cells. The epithelial cords is composed of two main categories of cells, chromophobes and chromophils. You can see that some cells are bright staining, it is chromophils, and some cells are pale staining, it means chromophobes. Chromophobes are more numerous than chromophils. Chromophils, it is hormone secreting cells, and they stain intensely, but not cytoplasm of the cell. In the cytoplasm present secretory granules in which hormones are stored, and especially these granules stains in different colors after hematoxylin staining. And we can distinguish two classes of cells with granules. Acidophils, cells who has pink or red granules in the cytoplasm, and basophils, cells which has the blue colored granules in the cytoplasm. Acidophils secrete simple proteins and stain intensely with eosin, also by the orange G, but respond negatively to the PES reaction. More abundant in the gland's periphery, it means that they lie on the periphery of the epithelial cords, and they are smaller than basophils, but their granules are larger and more numerous. Acidophils include two major hormone-secreting cell types. First, somatotrophs. Cell somatotrophs produce growth hormone, international abbreviation GH, growth hormone or somatotropin. The second group of cells produce prolactin and got name mammatrophs. Under the light microscope, it is very difficult to separate one type of acidophils from another, but it is possible under the transmission electron microgram. Somatotropin hormones, growth hormone or somatotropin, stimulate general body growth. It has influence on the epiphyseal plate in the bones. It is layer of hyaline cartilage between the epiphysis and diaphysis. Somatostatropin stimulates the growth of the bones, stimulating the proliferation of chondroblasts in this plate. The second hormone, prolactin, initiates and maintains the secretion of the milk after pregnancy. But without pregnancy, it stimulates the formation of the corpus luteum in the ovary after the ovulation, and also stimulate the production of hormone progesterone by the cells of this corpus luteum. Basophils secrete glycoproteins stained with hematoxylin and other basic dyes. They are usually larger than acidophils with fewer and smaller granules. Granules, I repeat once more, in these cells, basophils, stains in blue, not cytoplasm, but granules. Basophils can be subdivided into three groups. First, it is gonadotrophs. Gonadotrophs produce gonadotropin, but this gonadotropin may be different. In male, with its sex cycle, menstrual cycle, gonadotrophs produce different hormones according to the sex cycle. First hormone, follicular stimulating hormone, FSH international abbreviation. The other, in another part of the sex cycle, it produces luteinizing hormone, LH, luteinizing hormone. In males, this luteinizing hormone has name interstitial cell stimulating hormone, ICSH, international abbreviation, ICSH. And this hormone controls the activity of Leydig cells or interstitial cells, which produce hormone testosterone. The second group of basophils get name corticotrophs. These cells produce adrenocorticotropic hormone, ACTH, and this hormone controls the activity of the cortex of adrenal gland, of the suprarenal gland. Third group of basophils are thyrotrophs. Thyrotrophs produce hormone thyrotropin, thyroid stimulating hormone, or TSH. This TSH controls the activity of thyrocytes in the thyroid gland. It means that thyrotropin controls the production of the iodine-containing hormones of the thyroid gland. Now you see the light microscopic picture of the pars distalis of adenohypophysis. You can see the sections of the cross sections of the epithelial cords, one, second one, third one. Between them you loose connective tissue, delicate network of the loose connective tissue with the blood capillaries, sinusoidal sometimes, and fenestrated capillaries. It is larger magnification, the right part, it is larger magnification of the same microslide. And now you can clearly see the chromophils and chromophobes. On the left, you can see, for example, basophils. Especially gonadotrophes, it is not difficult to recognize. In these cells, nucleus locates acentrically. Now, for example, here it is basophils. You see blue granules in the cytoplasm and a centric nucleus. It is gonadotrophes. But if nucleus is in the center of the cell, like here, it may be thyrotrophes or somatotrophs. Or corticotrophs or gonadotrophs. Cells who has the red granules in the cytoplasm, you can see red granules in the cytoplasm here. It is acidophils. But we can't understand under the light microscope, is it somatotrophs or mammotrophs. Pale staining cells like here, it is chromophobes. Now you see the transmission electron micrograms. Here you see cells with granules, numerous large granules constitute the acidophils. And this very large magnification will show you the single acidophil cell with granules, dark staining, numerous large granules. Larger cell, it is basophils. And you can see that it may be gonadotrophs. Here, letter G, that's why author think that it is gonadotrophs. And he can be sure that it is gonadotrophs because in the cytoplasm of these cells we can find only few granules which are smaller than in the acidophils. We can see the section of the capillary, fenestrated capillary, in the between the epithelial cords. Chromophobes. It is group of small, weakly staining cells in the pars distalis. Generally, these cells has less cytoplasm and today we think that they may represent either non-specific stem cells or particular degranulated chromophils, although some retain secretory granules. That's why today we don't fully understand what is it chromophobes. No, some think that it is stem cell which can later, if it will be necessary, can differentiate into acidophils or basophils. The second hormone-active part of adenohypophysis is pars intermedia. It contains the Radke's cysts, which are small, irregular, colloid-containing cavities lined with cuboidal epithelium, and it is a remnants of Radke's pouch. It characterized by many cuboidal cell-lined, colloid-containing cysts, Radke's cysts. This picture shows you the Radke's cysts in the pars intermedia. On the right part of this picture, it is adenohypophysis. You see sections of the trabecules with the connective tissue around and many capillaries, fenestrated capillaries with the blood. Then we can see the sheets of epithelial cells, which are close, comparatively close, attached to one another. Between them is the layers of the connective tissue with the capillaries, blood capillaries, and these Radke's cysts. In the cyst inside, you can see the colloid. Cuboidal epithelium, you can see, covers the wall of the Radke's cysts. On the left part, it is neurohypophysis, pale staining region. Colloid of Radke's pouch rudimental Radke's pouch contains the Radke's cysts, and the content of the colloid in the Radke's cysts mostly consists of long molecule, which is secretory product of the epithelial cells of the cords. The name of this molecule, proopiomelanocartin, international abbreviation POMS. Proopiomelanocartin undergo the post-translational cleavage and is subdivided into 4 hormones: alpha melanocyte stimulating hormones, alpha MSH, international abbreviation, corticotropin, like corticotropin in the pars distalis, with the same function, then lipotropin and endorphin. Alpha melanocyte stimulating hormone stimulate the activity of the pigment cells, melanocytes, in the place of its allocation. It means this hormone controls the pigmentation of our skin, the color of our eyes, hairs, and some other regions. Corticotropin controls the cortex of the adrenal gland. Lipotropin, it is independent hormone who control the utilization of or metabolism of lipids which we eat. And if you ever saw the very thick person who has weight maybe 150 kilograms or more, very often it is deficiency of lipotropin, hormone lipotropin. And vice versa, if hyperproduction of lipotropin, this person become very thin, eat a lot of food, but don't become thicker. Now, today it is modern situation. and endorphin, hormone of pleasure and joy. This hormone controls our positive emotions, and its production is very important for our feelings. Very pity, but today endorphin is used for the creation of new synthetic narcotics, and this hormone is used synthetically changing endorphin today is used in many tablets. For example, hormones for discotheque, for aggression, for hormone which increase the physical activity of the person. And don't think that it is simple situation. According to the feedback mechanism, as I told you, if person eat endorphin in tablets, maybe atrophy of cells who produce this large molecule, proopiomelanocortin. And then if person stop to eat these hormones on the base of endorphin, will be deficiency of endorphin, and person can go to very hard depression. Now you again see the same picture like in the beginning of the lecture, correlation between the hypophysis and hypothalamus. As I said you, the nucleus of the anterior hypothalamus has multipolar neurosecretory neurons, which forms two nuclei, supraoptical and paraventricular nuclei. And axon of this neurosecretory neuron pass to the neurohypophysis, you see, and finish by the dilation. It means that neurohypophysis itself don't produce any hormones, but it has the dilation of axon of neurosecretory neurons of two nuclei of hypothalamus, and here in these dilations concentrates the hormones which produce in the bodies of these neurosecretory neurons. These hormones are two: vasopressin, or antidiuretic hormone, ADH in international abbreviation, ADH, and oxytocin. These hormones pass by the hypothalamus-hypophyseal tract, you see, and go to the neurohypophysis. Here they finished by the dilation, which get the name Herring's bodies. You see, Herring's bodies. And the main cell in which forms the neurohypophysis is pituitary. Pituitarides, it is modified glia-like cells which support these axons who travel to the neurohypophysis, and also covers these granule filled dilations, Herring's bodies. Then, according to the feedback mechanism, this antidiuretic hormone and vasopressin and the second hormone, oxytocin, go to the bloodstream according to the feedback mechanism. Antidiuretic hormone has influence on the kidney, and the vasopressin has influence on the smooth muscle cells in the organs where they present. Smooth muscle cells forms the wall of the uterus, myometrium. That's why this hormone has also influence on the tone of muscles in the uterus. And today this hormone is widely used for the stimulation of the labor, and this effect sometimes is very necessary for the pregnant woman. This microslide show you the Neira hypophysis, which is stained by methylene blue special dye. And if after the hematoxylinase staining, Herring's bodies are not visible, now they appear, they visualized, and you can see Herring bodies. It is spheric or ovoid bodies which are present in this Neira hypophysis. Around are pituitary cells, which occupy about 25% of the volume of the pars nervosa or Neira hypophysis. They are similar to neuroglial cells, as I said you, and help support the axon of the part nervosa by ensheathing them as well as their dilatations. Pituitary can contain lipid droplets, lipochrome pigment, and intermediate filaments. They have numerous cytoplasmic processes and forms the gap junctions with one another. Posterior hypothalamus is not clearly visible after the hematoxylin staining, and you can see this on the picture. Herring's bodies are not visualized. Again, the pars nervosa, and after hematoxylin staining, many components absent. Next organ which I want to tell you, it is the peripheral endocrine organ, thyroid gland. This gland anatomically locates on the trachea, on the front of the trachea, constitute two lobules and narrow isthmus between them. Embryonal development of thyroid gland start like exocrine gland. It begins during the 4th week of gestation from a primordium originating as an endodermal thickening of the floor of the primitive pharynx. The primordium grows caudally and forms a duct-like invagination known as thyroglossal duct. This duct descends through the tissue of the neck to its final destination in front of the trachea, where it divides into two lobes. During this downward migration, the thyroglossal duct undergoes atrophy. But in the beginning, the thyroid gland looks like exocrine gland with the excretory duct, which later obliterate. About the 9th week of gestation, endodermal cells differentiate into the plates of follicular cells that become arranged into follicles. During the 7th week, epithelial cells lining the invagination of the 4th branchial pouch, known as the ultimobranchial bodies, start their migration towards the developing thyroid gland. After fusing with the thyroid, ultimobranchial body cells disperse among the follicles, giving rise to parafollicular cells. Now you have to understand that thyroid gland is a combination of two glands. First takes place from the epithelium and forms the follicle, and the second, it is invagination of the 4th branchial pouch, ultimobranchial bodies, and these bodies fuse with thyroid gland and forms the second type of hormone productive cells in the thyroid gland, which get the name parafollicular cells. Under the light microscope, the thyroid gland is subdivided into the lobules, and this division takes place by the connective tissue septors. Gland is covered by the capsule, dense regular connective tissue, and capsule extend as septa into the gland. And now you see on this microslide the lobules of the thyroid gland. Around is the loose connective tissue or septus, which divides the organ into lobes and lobules. The smallest morphofunctional unit of the thyroid gland is follicle. Follicles, it is spheric or oval-shaped, sometimes irregular spheroidal structures. Its wall consists of one layer of simple columnar epithelium which rests on the basal lamina. And this columnar epithelium encloses a cavity that is filled by stiff gel called colloid. You can see colloid. The epithelial cells in the wall of the follicle may be of two types: principal or thyroid follicular cells. Historically, these cuboidal follicular cells has name thyrocytes. This name today is widely used. Thyrocytes. They constitute the majority of cells in the wall of the follicle. And between thyrocytes are dispersed single parafollicular cells or C cells. C cells. This picture shows you the section of the follicles. You can see colloid, oxyphilic, homogeneous colloid, and one layer of the cuboidal epithelial cells, thyrocytes, which derive from endoderm. In normal condition, these cells looks like this picture, cuboidal cells. But thyroid gland very often has pathologic changes, and mankind suffer from the diseases of the thyroid gland. The statistics says that it is not rare situation. If takes place the hyperfunction of the thyroid gland, in this case, these thyrocytes elongate, become taller, and looks like columnar cells. If takes place hyperfunction, these cuboidal cells change into flat cells. Hyperfunction means that cells become lower and show us hyperfunction. Now you can see larger magnification of the sections of the thyroid follicles. You can see the one layer of the columnar cells which rest on the basal lamina. Now it is adjacent follicle, one follicle, adjacent follicle. Now you see the cuboidal cells of the wall of the neighbor follicle. And between follicles maybe the thin plates of the loose connective tissue with the capillaries. This gland is very highly vascularized. Here we can see the fenestrated capillaries, and the secretory product of the thyrocytes go directly to the blood, to these capillaries. Transmission electron microgram show us that the follicular cells, thyrocytes, has a large nucleus which locates in the center, and cytoplasm has continuing numerous mitochondria, very well developed rough endoplasmic reticulum, and many lysosomes. Golgi apparatus and centrioles are located above the nucleus. And also between thyrocytes you can see other cells. It is C cells, which develops from ultra branchial bodies. These cells has granules, and you can see very well these granules which absent in the cytoplasm of thyrocytes. In the center of follicle is homogeneous colloid. In normal condition, this colloid is homogeneous. Colloid fills the follicular lumen. In fresh condition, it homogeneous, clear, and viscous. The main component of the colloid is thyroglobulin. Thyroglobulin, it is secretory product of thyrocytes. Also maybe enzymes in this colloid. Thyroglobulin, it is glycoprotein containing several iodinated amino acids. The main amino acid which undergo iodination, it is tyrosine. Now you can see again large magnification of the follicle with the cuboidal cells, and also you can see on the left part of the picture that maybe resorptive cavities in the follicle. When some content of the follicle colloid become pale staining. Usually it takes place in hyperfunction. That's why it is the picture of the hyperfunction thyroid gland, and you can see that cells are taller than on this picture. Very interesting secretory cycle is in thyrocytes. It involves the following steps. First, synthesis and storage of thyroglobulin. This protein is synthesized on the base of the tyrosine-rich protein, which is produced in the granular endoplasmic reticulum. Then it packaged in the vesicles in the Golgi complex, fused with the vesicles on the apical cell membrane, and then as a result of exocytosis go to the lumen of the follicle and forms the colloid. Next step, that amino acid tyrosine undergo iodination. It binds one molecule of the iodine and appear the monoiodotyrosine. International name MIT. The second molecule of tyrosine can bind two molecule or atom of iodine and appear diiodotyrosine, DIT. And the coupling of two DIT molecule forms tetraiodotyrosine or thyroxine. International name of tetraiodotyrosine, it is T4 or thyroxine. But when combined one diiodotyrosine and one monoiodotyrosine appear triiodotyrosine, T3, the second hormone of the which produced by the thyrocytes, where they take iodine. It appears as a result of oxidation of iodide. This molecule come from the blood, pump in the follicular cells, basal membrane, and they are transferred from the blood circulation. And this in the cytoplasm of thyrocytes these iodides cleavage into atoms of iodine, and these atoms of iodine combined with the tyrosine, amino acid tyrosine, in the cytoplasm of thyrocytes. Then these components are bind with the thyroglobulin and go to the colloid. Then, next step of secretory cycle, the receptor-mediated endocytosis. What does it mean? It means that thyrocytes has microvilli on its apical surface, and when it is necessary, according to the feedback mechanism, it endocytose this colloid back to the cytoplasm of thyrocytes. Then they liberate the T3 and T4 from thyroglobulin by lysosomal action. Ratio of T3 and T4 over 20 to 1. You have to understand that T4 hormone is less active than T3. And then, when they liberated from thyroglobulin, it can pass to the capillaries and reach the peripheral blood. So difficult and complex secretory cycle has thyrocytes, and maybe it is reason of so many pathologic changes in the function of the thyroid gland. The function of T3 and T4, they increase basal metabolic rate, then promote cell growth, increase heart rate, rise body temperature, generally enhance all energy requiring cell functions, are critical in the early stages of brain development and body growth. The deficiency of T3 and T4 in fetal period lead to oligophrenia, that not complete development of the brain. Now you see the scheme of the activation of the function of the thyroid gland. First, hypothalamus produce releasing or inhibiting hormones, thyrotroph cell stimulating hormone or thyrotroph cell inhibiting hormone. This hormone has influence on the pars distalis, on the thyrotroph cells. It is basophils, and this hormone, this cell stimulate the production of thyroid cell stimulating hormone, thyrotropin so-called. This thyrotropin has influence on the follicles of the thyroid gland, especially on thyrocytes, which increase or decrease the production of the T3 and T4 hormones. Then T3 and T4 hormones go to periphery and find the target cells with their receptors. That's why feedback mechanism can act in both, in all directions. If appear the positive stimul for thyroid gland from target cells, thyroid gland, increase the production of T3, T4. If negative, decrease. Same thyroid gland send the signal to the pars distalis, to the thyrotrophs, for stimulation or decreasing the production of thyrotropic hormone. And the same for hypothalamus. So this is the relationship between the hypothalamus, hypophyse, and thyroid gland. Now the second part of the thyroid gland, it is parafollicular cells. Parafollicular cells, as you remember, may be dispersed between the thyrocytes in the wall of the follicle. These cells are larger than principal cells, thyrocytes, has numerous granules, and produce hormone thyrocalcitonin, or simply calcitonin. These cells are not under the influence of the thyrotrophic hormone of adenohypophysis, independent, and act only on the basis of the feedback mechanism. Calcitonin lowers the blood calcium by causing calcium uptake by cells and increased calcium deposition in bone. So this hormone also produced by the parafollicular cells, which forms the clusters of cells between the thyroid follicles in the loose connective tissue. The second place of allocation of parafollicular cells as clusters of these cells in connective tissue between the follicles. In the first place, you can see it again, this transmission electron microgram. These cells are larger than thyrocytes. It is not visible here, but it is so, and their apical surface never reach the colloid. They are flat and large. One more transmission electron microgram, you see two parafollicular cells between the thyrocytes. You can see the apical surface of these cells never touch the colloid. Thyrocytes with microvilli, and capillaries, fenestrated capillaries around the parafollicular cells. Hormones go directly to the blood, and thyrocytes also hormone go directly to the blood. Next, peripheral endocrine gland is parathyroid gland. These glands usually are numerous. As you see the anatomic picture, they are located on the outer surface of the thyroid gland, in the laminas of capsule of the thyroid gland. Usually, person has several pairs of these glands, but maybe also accessory parafollicular glands, parathyroid glands, and that's why nobody can say how many parathyroid glands has concrete person. These glands develops from endoderm of the pharyngeal pouch, the superior parathyroids from the fourth pouch, and the inferior parathyroids from the third pouch. Each parathyroid gland is covered by the thin capsule, It consists of the sheet of epithelial cells and delicate septors which pass inward from the capsule, carry blood vessels, lymphatics, and a few nerve fibers into the gland. Between the epithelial sheets, now you can see the light microscopic picture after hematoxylinase staining. You can see these sheets of epithelial connective tissue, sheets of epithelial cells and plates of the loose connective tissue which start from the capsule. You can see connective tissue capsule which covers the gland. And you can see that this gland locates on the capsule of the thyroid gland. This is the section of the thyroid gland with its follicles and also septors and clusters of parafollicular cells between the follicles. In the epithelial sheets of the parathyroid gland present three types of cells. First of all, it is chief cells or principal cells. The second class, it is oxyphilic cells. And also maybe, today we know that also maybe intermediate cells. So principal cells are chief cells, also has name chief cells, and they are more numerous than oxyphilic cells. They have the basophilic pale staining cytoplasm, but has granules in the cytoplasm, blue granules. And these cells produce the parathyroid hormone, parat hormone, so-called parathyroid hormone, or simply parat hormone. This hormone is antagonistic to calcitonin of the thyroid gland, and this hormone increase the concentration of the calcium ions in the bloodstream. If calcitonin decrease the concentration of calcium ions and increase the mineralization of the bones, this hormone, parat hormone, increase the concentration of the calcium ions in the serum of the blood, and that's why decrease the mineralization of the bones. It is light microscopic picture after hematoxylinase staining of the parathyroid gland. You can see capsule, you can see cross sections of the sheet of epithelial cells, and delicate network of the reticular fibers as a plate of loose connective tissue. In the epithelial cords present, you see blue-colored chief cells, which are numerous, and they produce parat hormone. It is larger magnification, and you can see in details capillaries between the sheets of epithelial cells, a lot of basophilic cells or chief cells, and less oxyphilic cells with the pink cytoplasm. Oxyphilic cells are not hormone productive and don't produce any hormones. It is transmission electron microgram. You can see the oxyphilic cells which without granules and don't produce any hormones. Here around are basophilic cells. You can see the nerve fiber. Here it is nerve fiber, myelinated nerve fiber, and blood capillaries. A lot of blood capillaries are here. The parathyroid gland elaborate the parathyroid hormone, parat hormone, which responds to lower blood culture, and that's why it increases the reabsorption in the from the bone, from the kidney, and from the intestine. Increase the reabsorption of the calcium from in these organs. The last endocrine gland which we have and will be the last in my lecture, it is suprarenal gland. Suprarenal gland, it is paired glands which locates on the kidney or so-called adrenal gland. They look pelvic region. You can see it is pelvic bones, it is kidney, and on the kidney, on the top of the kidney locates the adrenal gland or suprarenal gland. This gland on the section has the outer portion which called cortex and the inner component medulla. Embryonic development of this organ, that cortex, develops from coelomic intermediate mesoderm, and the medulla has another origin. It derived from the neural crest and presumptive autonomic ganglion tissue. So the cells of this tissue are pale staining after hematoxylination because they are modified glial cells and produce mediators of neural system. But cortex, as you understand, it is derivative of mesoderm from, that's why it is epithelial tissue. The cortex of the adrenal gland has 3 zones. Just under the capsule, blue it is capsule, under the capsule locates the zona glomerulosa. Something here you can see epithelial cords, and these epithelial cords forms the loops just under the cortex, which looks like glomerulus. Then these sheets of epithelial cells travel parallel to one another and appear the zona fasciculata. Zona fasciculata. And the third zone, it is the zona reticularis, when the regular allocation of epithelial cords change and cells forms reticulum. Ratio between these zones 1 to 9 to 3. This picture shows you zona reticularis. It is lower, it is part of the medulla. Zona glomerulosa is the most narrow zone immediately beneath the capsule, constituted of pyramidal or columnar cells arranged in ovoid groups or arcades that are continuous with the cell columns of the zona fasciculata. These cells has lipid inclusions, a lot of lipid droplets, and produce lipid-soluble hormone, mineralocorticoid. It is a family of hormones, mineralocorticoids, steroid hormones, lipid soluble. The main hormone principle has name aldosterone, and also produce some deoxycorticosterone. This hormone, aldosterone, control the water and electrolyte balance mainly by stimulating sodium absorption by the renal distal tubules. Later we will discuss this hormone again when we will study the urinary system and kidney. Next zone, zona fasciculata. It is the thickest layer of the cortex, is composed of large irregular cuboidal or polyhedral cells arranged in long radial cords, usually 1 or 2 cells wide. The cords are separated by sinusoidal capillaries from one another. Nuclei of cells of this zone are located centrally, and in the cytoplasm we also can find a lot of fatty acids and natural fat and lipid droplets. These cells of zona fasciculata secrete another lipid soluble hormone, steroid hormone, a family of glucocorticoid hormones. The main are cortisol and corticosterone. The synthesis of this hormone is stimulated by adenocorticotropic hormone of the adenohypophysis. Also, this hormone has influence on the zona glomerulosa and have no influence on the zona reticularis. That's why glucocorticoids, its production controlled by the hypophysis. Glucocorticoids today are widely used in medicine, and today pharmacology enterprises create every day work with these hormones and create the new synthetic glucocorticoid hormones. Some of them are more active and stronger than native in many times. The main function of glucocorticoids: control of fat and protein metabolism, control carbohydrate metabolism, suppress the immune response by decreasing circulating lymphocytes and eosinophils, suppress the formation process. You see how fruitful is these hormones for medicine, for therapeutics, and also for surgery and other branches of these main disciplines. Zona reticularis. The cells of this zone, it is the innermost cortical zone, and this zone form a network of anastomosing cords that also are separated by sinusoidal capillaries. These cells are smaller than in other zones, especially in zona fasciculata, and their cytoplasm also has lipid droplets, but not so numerous like in zona fasciculata. These cells of this zone secrete sex hormones, androgens like dihydroepiandrosterone and estrogens. First, it is analog of testosterone, male hormone, and estrogens, it is female hormones. But very interesting that zona reticularis secrete hormone of the opposite pole. In women, they secrete androgens, and in men, the zone secrete estrogens. So these cells Also can secrete and synthesize a small amount of glucocorticoids, but it is not critical. This production of these hormones is not controlled by the adenocorticotrophic hormones. And some words about medulla. As I said you, that medulla develops from neural crest cells, has the neural origin. It is modified glial cells who are large, ovoid, or polyhedral, occur in groups or short anastomosing cords surrounded by venules and capillaries. Two main types of cells, chromaffin and ganglion cells. Hormone productive are chromaffin cells. Why chromaffin? These cells, like all components of the nerve tissue, has better stained by salts of the heavy metals. Now salts of chrome stains these cells in black color. That's why chromaffin cells of the medulla. There are two types. You have to know at least two types: epinephrocytes and norepinephrocytes. First, epinephrocytes produce adrenaline, norepinephrocytes noradrenaline. So this medulla of the adrenal gland produce the mediators of the neural system. And this zone of this organ provide the stress reactions. If person feel the emotion stress, temperature stress, infections, maybe also a stress factor, and some other mechanical stress, pain stress. In this case, chromaffin cells start to produce a lot of adrenaline or noradrenaline, depends from the reason of this stress, and it provides the stress reactions. So doctors, thanks for your attention. My lecture is over and you may be free. Goodbye.